Welcome back. So we are going to finish the second part of our energy and enzymes lecture today. And recall last time we had finished talking about central metabolism. We had finished talking about all of our different enzymatic reactions, right? Hydrolysis and dehydration, redox, exergonic, endergonic, and isomerization. Took me a second there. I couldn't quite remember them myself. So here we're actually going to zoom in real close and look at how individual enzymes work together. So the basic idea behind how chemical reactions happen is just molecules bumping into each other. Molecules react with each other by randomly bumping into each other in the right orientation, so at the right angle and at the right speed. And it takes some effort in order to get molecules to bump into each other in the right way. It takes time, it takes effort. And what enzymes do is they simply catch those molecules, they properly orient the molecules, and make sure they come together in the right way the first time. So enzymes are kind of like dating sites for molecules. They make sure that things happen the right way, right, in the right order. Maybe that's a bad analogy because dating sites don't actually work that way, but you get the idea. So not only do enzymes catch and orient molecules, they may supply the energy needed for those molecules to actually do something, for those molecules to actually rearrange their chemical bonds, to donate electrons to each other, etc. Now the thing about chemical reactions in general, not just enzymatic reactions, but all chemical reactions, is that they increase with temperature. The higher the temperature, the faster molecules are moving. So we call the natural movement of molecules Brownian motion of molecules. That's not a term you need to know, but if you'd like to see a simulation of molecules moving, you can go ahead and click on that. It's a YouTube video. And the higher the temperature, the more Brownian motion. So with enzymes, same thing. The higher the temperature, the easier it is for enzymes to do their job. Now, you may be going, well, wait a second. Sometimes those enzymes denature, right? And I just realized I needed to put this full screen. Sometimes those enzymes denature, and you'd be correct, right? The higher the temperature is, the better, or the faster chemical reaction goes up until the enzyme denatures. So here's what enzymes do. They take a chemical reaction. Here we're taking A plus a molecule that's been joined together, B and C. We're rearranging the chemical bonds in these two molecules and we're producing AB plus C, right? Our reactants are over on the left, products are over on the right. This is actually an exergonic reaction. This is an exergonic reaction. All right. And what an enzyme does is it takes this energy of activation and it lowers it. It lowers it. Add a device. Tap to set up your pen. No, I don't need to add a device. All right. An enzyme takes the energy of activation and lowers it. So it takes the time and effort it takes to properly orient those molecules and decreases it. Because it decreases it, it's easier to get over this energy of activation hump, and a reaction will proceed faster. So here we see an enzyme catalyzed reaction where that energy of activation has shrunk. So just to kind of flicker back and forth, let me erase my terribly drawn in line here. All right. So before an enzyme, after an enzyme. Before and after. Before and after. And in reality, I mean, this is obviously a simplified version. In reality, here's what the action actually looks like before and after an enzyme. Here's before an enzyme. There's the energy of activation. And here's with an enzyme. An enzyme drastically decreases the energy of activation, not just by a little bit, but by a huge amount. All right, so we, of course, have special terms for how enzymes work. And so let's review some of that basic enzyme vocabulary. The reactant that an enzyme acts on, so in the previous image, it's this A plus B and C. Those are our reactants. We don't just call them reactants, we call them substrates. We call them substrates. So an enzyme substrate is what it reacts on. So let me draw a little picture down here. Here's my enzyme. I'm going to draw him looking like a Pac-Man. And this little area right in here, I'm going to highlight in red. This is the area that a substrate will bind to. Alright, so let me draw my substrate in blue. Alright, so substrate. As soon as a substrate binds to or attaches to that special area of the enzyme, we have a big group of enzyme and substrate. We call that the enzyme-substrate complex. And that area that I highlighted in red previously that actually binds to the substrate, that's called the active site of the enzyme. So that's the part of the enzyme that actually attaches to the substrate. Now what happens is as soon as a substrate attaches to an enzyme, remember, way back when, whenever you add anything to your protein, especially something as big and bulky as a substrate, whenever you add anything to a protein, you change the protein's shape. So as soon as you add a substrate to an enzyme, that enzyme will change shape, and the way it changes shape is actually to kind of to grab on more tightly to its substrate and bring some of its reactive R groups on its amino acids into contact with the substrate. Remember, individual amino acids, they have one of 20 possible R groups. So by doing induced fit, by changing its own shape, the enzyme will bring some of its very reactive R groups into place, and it'll allow those R groups to actually start catalyzing chemical reactions, pushing electrons, pushing energy. So here we see this. Here's an enzyme, an empty enzyme. We can see the active site highlighted. Our substrates are binding, right? And then we form the enzyme-substrate complex. Notice the enzyme has changed shape. It's undergone induced fit. Because of that induced fit, the enzyme will be able to catalyze the rearrangement of energy and electrons. Alright, so we convert those substrates into products. We don't have any special name for the products, we just call them products. The enzyme will then release the products and go back to its original state. So enzymes can work over and over and over and over again without being used up in the process themselves. So we said that we have a unique enzyme for almost every chemical reaction in our cells, right? Well, how do enzymes know what substrates they're supposed to act on? Well, it's actually based completely off of shape, right? So here's an enzyme right here. This is a slightly different form of the enzyme. We see all the individual atoms and molecules here. This is our good buddy hexokinase. Remember, he was the one that took glucose and turned him into glucose 6-phosphate. Right? And along the way, he took some ATP and hydrolyzed it into ADP, an inorganic phosphate. Alright? So when we look at hexokinase, there's a specific area within the hexokinase, the active site, where both glucose, the substrate, and ATP, another substrate, will fit. And only glucose and only ATP will fit. So here we see that enzyme kind of curling around the glucose and ATP. That has undergone induced fit. Alright. So the shape of the substrates must fit into the active site of the enzyme. And that's how enzymes recognize their substrates. Everybody has a unique shape. So this is what the active site of an enzyme actually looks like before it's folded and after it's folded. So here we have the primary structure. Remember, our protein structure is primary, secondary, tertiary, quaternary, and primary structure is simply the line of amino acids all linked together. This denotes one end and this denotes the other end of the protein. So here, when this enzyme folds fully, it's only certain amino acids that will actually end up in the active site. It's these purple guys right here. So when this enzyme fully folds, all those purple guys will end up close to each other, forming the active site. And the way these purple amino acids work, and remember these purple amino acids, each one of them has their own unique R group. These R groups usually hydrogen bond to the substrate. So very often, these amino acids, because they have to hydrogen bond, they have to be hydrophilic. Remember, if you're hydrophilic, you are capable of hydrogen bonding. These R groups that end up in the active site are usually going to be hydrophilic. All right. So later on, you may hear about certain mutations that change a hydrophilic R group into a hydrophobic R group, right? That is really, really devastating if it happens in the active site of a particular enzyme. So here's a really cool example. All right, so this is an enzyme named alcohol dehydrogenase. Alcohol dehydrogenase. Based on the name, dehydrogenase, you should already be able to guess, that he probably does a redox reaction. I keep doing that. He probably does a redox reaction. All right. So this is his shape. Here we see the ribbon model of this enzyme. We don't see all the individual atoms here. All right. I don't actually know where his active site is on here. It's somewhere in there. Alcohol dehydrogenase normally acts on alcohol and oxidizes that alcohol. Well, the thing is this alcohol dehydrogenase's active site is a little loosey-goosey here. All right, so not only can it recognize alcohol, it can also recognize this other molecule that kind of looks like alcohol. This other molecule is called ethylene glycol. It's also known as antifreeze. This is the stuff you put in your car. So if you were to drink antifreeze, not recommended by the way, please don't do this. If you were to drink antifreeze, this enzyme, alcohol dehydrogenase, would take antifreeze, or ethylene glycol, and oxidize it into something poisonous for you. So I'm going to draw a little skull and crossbones. It's a crappy little skull and crossbones, but you get the idea. All right. So when we break down antifreeze, it actually produces a poison. This is why you should not drink antifreeze. Well, the thing is, if you drink antifreeze on accident, or maybe you have a pet who drinks antifreeze, or maybe you have a kid who drinks antifreeze, and I know you're going, why in the world would they ever drink antifreeze? Well, the thing about antifreeze, it's bright blue and it's slightly sweet tasting. So kids and pets tend to love it. So if you have any spilled antifreeze on the floor of your garage, you gotta be careful. The thing about antifreeze and alcohol is if you drink a ton of antifreeze, what you can do in order to counteract the toxic effects of the antifreeze is you can actually drink a ton of alcohol. So, the idea here being that if you flood this enzyme with equal amounts of both antifreeze and alcohol, well, the enzyme will act on either one, right? It'll slow down the rate at which you oxidize antifreeze because you flooded the enzyme with alcohol. You'll produce this poison at a slightly lower rate, and you are better able to metabolize that poison away before it has the opportunity to kill you. So this is absolutely not recommended, but if you find yourself in an area where you can't get to a hospital very easily, or you can't get to a veterinary clinic very easily, and you call poison control, they'll say, give them the highest proof alcohol you can find. But again, if you have a choice, go to the hospital. Don't chug some booze after drinking antifreeze, okay? Okay, so what else affects enzymatic activity? Remember, enzymes are proteins, so the same things that impact proteins are the same things that impact enzymes. Heat, pH, salt concentrations, all of these will affect enzymatic activity. But in particular, we have a couple other things. So one class of other molecules that affect enzymatic activity are called cofactors and coenzymes. Cofactors and coenzymes. Cofactors are small ions like magnesium and zinc and iron They help enzymes fold properly. These ions are highly charged, so they can help enzymes fold. They also assist in donating or accepting electrons during redox reactions. Especially iron. Iron is really good at this because iron has multiple oxidation states, as you may remember from chemistry. Coenzymes do the same thing. They help enzymes fold properly. They also will assist in redox reactions and certain other things too. We're not going to get beyond this and what those certain other things are. So cofactors and coenzymes help enzymes work properly. They are good. Competitive and non-competitive inhibitors, on the other hand, are things that slow enzymes down or stop them. So a competitive inhibitor will bind to the active site of an enzyme and prevent the normal substrate from binding. So actually, over here, if we were to treat antifreeze poisoning using alcohol, alcohol would be considered the competitive inhibitor. A non-competitive inhibitor binds somewhere else on the enzyme, causes the enzyme to change shape, so the enzyme is no longer able to bind to its substrate. So both these inhibitors do the same thing. They prevent the enzyme from attaching to its substrate, but just in different ways. Okay, so a little bit of background information here. The average rate of an enzymatic reaction is about 1,000 reactions per second. These guys are insanely fast. So the fastest enzymes are something like 5,000 reactions per second. The slowest enzyme is actually an enzyme in photosynthesis. It's 3 reactions a second. So to give you an idea of why this is so fast, if a substrate for a particular enzyme is 1 in every 105 molecules in a cell, that's a bit of a high concentration, but just theoretically speaking, it was 1 in every 105 molecules in a cell, an enzyme will encounter its substrate 500,000 times a second. So it's not at all surprising that these enzymatic reactions actually happen quite quickly. Now, just because we have plenty of substrate present doesn't mean the enzyme will automatically work at its optimal rate, right? Temperature, pH, how tight that active site fits to the substrate, so the natural affinity of the active site for the substrate, and how much substrate there is all affect enzyme activity. Enzymes are what we say saturable. You can add substrate and they will increase their activity, add substrate, they'll increase until you just overwhelm them. There's too much substrate, too little enzyme, they'll reach their maximum capacity. So we say enzymes are saturable. So when we first add substrate, enzymatic activity will increase, it'll increase until it finally levels out, it flattens out. So here, we can actually see this. I'll go back to the previous slide in just a second. We can actually see this. Here's an uncatalyzed reaction, just some generic reaction. We can see that it's pretty slow, especially compared to an enzyme catalyzed reaction. And notice when we first start adding the substrate, we really pick up speed, and then we continue adding substrate, but we level off. So after a while, we max out the capability of our enzymes to do that chemical reaction. Now what we could always do is we can just add more enzyme, and this curve will keep going up, but that takes time and effort from our cells. Not only are enzymes saturable, they are affected by temperature and pH. So here we actually see enzymes from 2 different organisms. The blue line is an enzyme from the same enzyme, by the way. We're looking at the same enzyme in these 2 organisms called chitinase. This blue line is from a bacteria that lives in a cool environment, all right, and a neutrally charged environment. And we can see that the enzyme's activity peaks, right around 47 degrees Celsius, which is still pretty warm. This is from a bacteria that lives in a hot and acidic environment. We can see its particular enzyme's activity peaks around, 60 degrees Celsius. Similarly, that bacteria that lives in the neutral environment, his enzyme's activity will peak around pH 7. The one that lives in the acidic environment, this enzyme's activity will peak at around pH 3 or something. So, what determines whether an enzyme, where an enzyme has its peak activity? It's all about the particular sequence of amino acids that it has. So if you change around the amino acids, you change the patterns of bonding within the protein, within the enzyme, and you change when it's, whether it's very, very stable in hot temperatures or whether it's very not unstable. You change whether it's very stable in acidic temperatures, in an acidic pH, or whether it's more stable in a neutral pH. It all depends on the particular amino acids that are in the enzyme. All right. So, going back, these cofactors, coenzymes, are another major way that we can impact enzyme activity. In addition to cofactors and enzymes, these are the two main ways that we can increase enzyme activity. We also have, kind of on the side here, we also have these things called prosthetic groups. So let's look at some examples of cofactors, coenzymes, and prosthetic groups. Assuming that I can actually get this to go forward. There we go. Okay. So these cofactors, coenzyme, and prosthetic groups, they're not really considered part of the enzyme itself. They can detach and reattach and detach and reattach, and very often they have to be produced separately from the enzyme. So enzymes are made through transcription and translation. These cofactors and coenzymes either have to be eaten or synthesized through a totally separate metabolic pathway. So as we said before, cofactors are inorganic ions. They include mostly metal ions like zinc, magnesium, and iron. And they reversibly interact with enzymes. We said that they can bind, then detach, then bind, then detach. They help enzymes fold properly, and they can help substrates attach to the enzyme. They also help with redox reactions. Coenzymes are organic molecules. They're usually a couple dozen carbons. They're not really considered a protein or a nucleotide or any one of our traditional biological molecules, although we can convert proteins and nucleotides and lipids into coenzymes. They do the same thing as cofactors. Right? They reversibly interact with enzymes to help with proper folding, substrate stabilization, and redox reactions. Coenzymes include our vitamins. So, remember we talked about vitamin B2 and vitamin B3. They actually become NADH and FADH2. And we said, well, the job of coenzymes is to help with redox reactions. So remember, NADH and FADH2 are the electron carriers for our redox reactions. So these are our two most important coenzymes, NADH and FADH2. Things like vitamin C is also a coenzyme. Vitamin A is a coenzyme. So as an example, let me go back here after I accidentally flipped to the next couple slides. There we go. Vitamin C and vitamin A are both necessary for certain enzymatic reactions to take place. Vitamin C is necessary for the enzyme that produces collagen. So if you don't have vitamin C, the enzyme that produces collagen doesn't work properly. Well, that enzyme, if it doesn't work properly, you get no collagen. And you guys may know that if you don't get enough vitamin C, it results in a disease called scurvy. Well, the symptoms of scurvy are all associated with the lack of collagen. We find collagen in our skin. Collagen is what holds our teeth in place, right? So if you don't properly produce collagen, your teeth are going to fall out. Your skin starts to bruise really easily. You can't hold your blood vessels in place, right? Your bones become super brittle as you can't produce collagen. Your joints become super flexible because that collagen that holds them together starts to degrade. So all the symptoms of scurvy. Vitamin A is also another coenzyme that's necessary for your eyes. So in the retina of your eyes, the very, very back of your eyes, all right, the very, very back of your eyeball, right where your optic nerve comes out, you have a layer of cells called the retina, and those cells have enzymes that need vitamin A to function properly. So vitamin A is actually used to register light. So if you have no vitamin A, you can't register incoming light, which means you can't see. Without vitamin A, you are blind. All right, prosthetic groups are a third type of enzyme behavior modifier. They're kind of like, they're what we uh, so if it's not a cofactor, if it's not a coenzyme, it's a prosthetic group. They're kind of this big bucket into which we throw everything that we don't know how to classify. They're non-amino acid molecule atoms or molecules. But the thing is, what's unique about them is that they are usually permanently attached to proteins. They're permanently attached to proteins and perform essential functions. They do a lot of different things. Let's look at some examples of these cofactors, coenzymes, and prosthetic groups. So, right here, this molecule is called hemoglobin. Hemoglobin is what allows you to carry oxygen. It actually binds to oxygen in your lungs and carries oxygen throughout your body. And hemoglobin is made of a combination of protein. So here we see the alpha helices of the protein. It's made of the prosthetic group heme, and it's also made of the cofactor iron. So we have iron in there as well. So an enzyme can have a cofactor, a coenzyme, and a prosthetic group, just a cofactor, just a coenzyme, just a prosthetic group, or any combination of the three. Hemoglobin has a prosthetic group and a cofactor. Here's our good buddy, DNA polymerase III. Here's the DNA molecule that he's acting on right there. I'm highlighting him in green. And remember, there's the exonuclease domain. That's the one down here in gray. Remember, that's the one that does the proofreading. Well, DNA polymerase, in order to function properly, he needs magnesium ions. So that red ball right there is a magnesium ion. One of the things we do during our Sanger sequencing reaction, in lab, is we actually have to add magnesium ions in order to make sure that our DNA polymerases work properly in order to do our Sanger sequencing reaction. All right, here's one more example. So magnesium is by the way, is a cofactor. Here's one more example of another cofactor, zinc. So we need zinc. There are these little green balls in here. We need zinc in order to allow our transcription factors to fold properly and actually bind to DNA. So zinc is part of a tertiary motif called a zinc finger. We find zinc fingers a lot in transcription factors, and we need those zinc fingers in order to properly attach to DNA and regulate gene expression. All right, let's look at our inhibitors now. So, just to keep us organized, going back, all the way back, remember we have two major ways of regulating enzyme activity: cofactors and coenzymes, and inhibitors. So we just covered cofactors and coenzymes. We are looking at inhibitors now. Okay. So with inhibitors, our two major types of inhibitors are competitive inhibitors and non-competitive inhibitors. We also have a third type called allosteric regulators. They're not really considered a true inhibitor, and we'll see why in just a moment. All right. So let's look at our competitive inhibitors first. A competitive inhibitor is a molecule, and this can be any sort of molecule. All right. This molecule binds to the active site of an enzyme, so essentially blocks the enzyme, the active site, prevents the substrate from binding. All right. And there are two types of competitive inhibitors. There are those that are reversible. Their binding is very temporary, right? attach, then detach, then attach, then detach, then attach, then detach. And then there are the irreversible competitive inhibitors. They actually covalently attach themselves to the enzyme. Once they're attached to the enzyme, that enzyme is out of action. Non-competitive inhibitors, on the other hand, are molecules that bind to another part of the enzyme, changing the enzyme shape and preventing the substrate from binding. They are also reversible, categorized into reversible and irreversible. And here's where the terminology gets a little wonky. A non-competitive inhibitor is the same as a negative allosteric regulator, which we'll see what those are in just a second. So just keep that in mind, all right? So let me go on. Let me look at competitive and non-competitive inhibitors. Here's a normal enzyme right there with its normal substrate living its normal life, all right? The substrate fits into the active side of the enzyme based on shape. A competitive inhibitor will block the active site, so this enzymatic reaction doesn't take place. A non-competitive inhibitor will bind elsewhere on the enzyme, change the shape of the active site so the substrate can't bind. So let's look at our allosteric regulators now. An allosteric regulator is any molecule. It can be another protein, it could be a small carbon chain, right? It could be any molecule. All these can be any molecules. That's why there's a little star next to them. This molecule binds to a separate part of the enzyme besides the active site, and it changes the shape of the enzyme. Now here's the thing about allosteric regulators. By the way, allo means at a distance. So that's why it binds to another part of the enzyme. We can have negative allosteric regulators. These decrease or halt enzymatic activity, and these are actually the same as this is the exact same thing as a non-competitive inhibitor. All right. So a negative allosteric regulator binds to another part of the enzyme and decreases the enzyme's activity. That's the exact same thing as a non-competitive inhibitor. A positive allosteric regulator will bind to another part of the enzyme and increase the enzyme's activity. Phosphorylation is actually a form of positive allosteric regulation. Phosphorylation changes the enzyme shape and activates the enzyme. And usually the way it'll activate an enzyme is it'll open up the active site. All right, going back to negative allosteric regulation is often used during a process called feedback inhibition. We'll see that in just a few slides. Okay, so leaving behind the feedback inhibition for a moment, let's look at some examples of inhibitors. Okay, this is where it gets really cool. So when we talk about competitive and non-competitive inhibitors, this is where we really bring pharmaceuticals into play. So those of you guys who are studying pharmacy, or those of you guys who are thinking about going into medicine, you're going to be talking a lot about enzyme activity and a lot about competitive and non-competitive inhibitors. In fact, if you ever go to the pharmacy and like you pick up a new medication, they give you that that tiny little piece of paper that's folded up really tight. It's folded up like a map, and then you unfold it and it becomes huge, and there's tons of tiny little text on there. Well, if you read that text and look really closely, it'll actually describe exactly how that medicine works. And a lot of medicines actually work as competitive or non-competitive inhibitors. So it'll say on there, on that tiny little piece of paper somewhere, that that particular medication is a competitive inhibitor of a certain enzyme. So let's look at one particular medicine called statins. Statins are a class of drug that lower cholesterol. So some examples of statins include Lipitor, Lescal, Crestor, Zocor. They all kind of have these similar sounding names. And they competitively inhibit an enzyme called HMG-CoA reductase. So HMG-CoA reductase is down here in this long metabolic pathway. HMG-CoA reductase takes this molecule, HMG-CoA, and converts it into this molecule, mevalonic acid. So that mevalonic acid will eventually go on to produce cholesterol. Alright? So if we inhibit HMG-CoA reductase, knock him out of the equation here, we can no longer produce mevalonic acid, and we can no longer produce cholesterol. So statins are actually similar in structure to HMG-CoA. They're similar in shape to this HMG-CoA reductase's normal active site. Makes sense. You would want to make a drug that's similar to the, just say normal active site, you would want to make a drug that's similar to the enzyme's normal substrate in order to produce a competitive inhibitor, because they both got to fit into the same active site. Here's another example. Okay, actually before I go on, one more thing here. This is actually a reversible competitive inhibitor right here. So most statins are reversible competitive inhibitors. Here's an example of an irreversible competitive inhibitor. Alright, this is how aspirin works. So let me give you the metabolic pathway behind pain, because aspirin relieves pain. So when our cells are damaged, obviously that's when we register pain, our cells will convert a molecule that they have floating around inside them called arachidonic acid. They'll convert arachidonic acid into something called prostaglandin. The enzyme that does this conversion is called COX-2, stands for cyclooxygenase 2. So prostaglandin is actually a cell signaling molecule. It signals cells to undergo inflammation, to increase the internal thermostat, so increase heat, and to signal for pain. So if we inhibit this COX-2 enzyme, then we stop these processes. We stop pain signaling, we stop inflammation, we stop the increase of that internal thermostat. So right here, this is how the COX-2 enzyme works. So this is the full COX-2 enzyme right there. This is aspirin right here, and let me actually, here's aspirin, I'm highlighting it in green. Okay, aspirin, and aspirin. And aspirin's going to attach to the active site of the COX-2 enzyme. Here's the just one amino acid out of that COX-2's active site. That one amino acid, the one amino acid that I'm drawing in purple here. Alright, so aspirin attaches itself to the active site of COX-2, and specifically it attaches itself to the amino acid serine. It'll chemically modify the serine. And those of you guys who know your organic chemistry, you can actually follow these little arrows. These make sense. They follow the laws of organic chemistry. And we'll take that serine, which formerly had an OH, and we'll convert it into this into an acetylated serine. So remember, an acetyl group has a C double bond OCH3. Alright, so we've chemically modified that serine and prevented that serine from working properly within the active site of COX-2. So that COX-2 enzyme is knocked out of action, and in order to trigger inflammation and pain, our cell just has to produce more COX-2 enzyme. And that will take several hours, which is why aspirin works for several hours. Now, interestingly, if you overdose on aspirin, you don't need to know this. If you take too much aspirin, it also will it'll affect other enzymes besides just this COX-2 enzyme. It'll disrupt the electron transport chain. In the electron transport chain, instead of putting its energy towards making ATP, it'll just waste that energy and release the energy as heat. So an overdose of aspirin will actually trigger a fever. Okay, a couple others. And these you do not need to know. This is just for, this is cool, we bothered to learn all this stuff, now let's apply it sort of deal. Alright. So proton pump inhibitors, these include Prilosec and Nexium. These help to treat heartburn. How they work is they are actually competitive inhibitors of acid pumps in the stomach. So heartburn is where you overproduce acid in the stomach, and that acid crawls up your esophagus and produces that feeling of heartburn. Prilosec and Nexium are actually competitive inhibitors of those acid pumps. So less acid equals less heartburn. Selective serotonin reuptake inhibitors, on the other hand, oftentimes we just shorten that name to SSRIs. Selective serotonin reuptake inhibitors actually help to increase serotonin concentration in synapses. So, do I have it? No, I don't. Okay. Let me draw it over here. So here we have, let me draw two neurons. Here's the tail end of one neuron, and here's the beginning of another neuron. So there's this area between the two neurons. It's a crummy looking neuron, but that's okay. This area between the two neurons is called a synapse. Dang it. This area between the two neurons is called a synapse. And a neuron will release neurotransmitters into that synapse. So I'm going to draw in my neurotransmitters in green. And those neurotransmitters will attach to receptors on the next neuron. Well, one neurotransmitter that we often use is called serotonin. All right. And one of the many possible things we think could cause depression is a lack of serotonin, where we're not releasing enough serotonin into that synapse. Serotonin is a neurotransmitter that makes you feel content, it makes you feel happy, it makes you feel calm. Right? So if you don't produce enough serotonin, you don't really feel these things, and that may result in depression and anxiety. And it's important to note that there are lots of causes for depression and anxiety. This is just one possibility. All right, so when we release that serotonin, we don't want to waste it. We're going to recycle it. So the neuron that released it is going to suck up that serotonin through these little pumps, all right, to be able to reuse it again. So what antidepressants like SSRIs do is they block these little pumps from working, so that serotonin that's released spends more time in the synapse. And this cell that's receiving the serotonin, thinks he's getting more serotonin than he actually is. So because he thinks he's getting more serotonin than he actually is, right, this actually may treat the symptoms of depression, which is a lack of serotonin. So drugs like citalopram, also known as Celexa, Prozac, sertraline, also known as Zoloft, all work this way. They block the serotonin reuptake pumps. All right, our last one is called angiotensin converting enzyme inhibitors. These are, angiotensin is a signaling molecule, and it causes increased blood pressure by increasing the amount of salt and water that you have in your blood vessels. So if you have more salt and water, that's more pressure on your blood vessels, you increase your overall blood pressure. So these actually treat high blood pressure. So these are competitive inhibitors of angiotensin production. We normally produce this angiotensin in the liver. So if we stop the production of angiotensin, we stop increasing the amount of salt and water in our bloodstream, we lower our blood pressure. So lisinopril is a major drug that we use for treatment of high blood pressure. Capitin is another one. And they are just competitive inhibitors of the enzymes that produce angiotensin. By the way, if you guys, while I'm thinking about it, this entire class of enzymes are called ACE inhibitors, or this entire class of drugs are called ACE inhibitors, angiotensin converting enzyme inhibitors, if you want to look those up. Okay, so this all brings us to this idea of feedback inhibition. You don't just want to let your metabolic pathways run like crazy, right? You don't want to let them run without regulation. So we have to have a way to regulate our metabolic pathways, and the major way we do it is through a process called feedback inhibition. So here we see a metabolic pathway right here. This is a metabolic pathway. Here we're converting a substrate into intermediate using enzyme 1, intermediate A into intermediate B using enzyme 2, intermediate B into product using enzyme 3. We're familiar with this by now. It may be a catabolic pathway, it may be an anabolic pathway, it doesn't matter right now. Okay? So here's the thing. If this metabolic pathway is going for a while and you're building up lots and lots and lots of product, well, maybe you go, hey, I got plenty of product. Let's save some of that substrate for a little later when I may not have as much substrate. So what we will often do is we will use this product to act as an allosteric regulator of the first enzyme in this pathway. So this product will come back, it'll bind to this first enzyme, and it'll cause that enzyme to shut down. So it's actually, most of the time, it's going to be a negative allosteric regulator. This process of producing a product and that product traveling back to shut down that first enzyme is called feedback inhibition. Feedback inhibition. So this shuts down a pathway when products are no longer needed by a cell. Alright. So that is it for enzymes. Now the nice part of this is that we've done all the heavy lifting for cell respiration. So this is the hard part. This is the hardest part, I think, of our metabolism lecture section. When we look at cell respiration now, we've learned about all the different types of chemical reactions. We've learned about redox reactions. We've learned about NADH and FADH2 and ATP. All we got to do is put them together. So as always, thanks for sticking with me, guys, and I will see you next time around.